So the paper was on the design of orthogonal genetic switches based on a crosstalk map of sigmas, anti-sigmas, and promoters. And to really understand or grasp the paper, you first have to know a lot of the terms. So when they mention orthogonality, they mean factors that can be used in circuits without interfering each other. So if there is one sigma factor, if there are two sigma factors being in place, they will not interfere and they won't affect each other's cognate sigmas. So then the question is, what is a sigma factor? They're basically these promoter regions that are on the RNA polymerase. So usually the RNA polymerase is sort of free-floating and it's going around everywhere. But in order to bind to a particular region, it needs the sigma factors. And the sigma factors can be stress activated, but they also they have particular domains that they recognize, and that's how the RNA polymerase knows where to bind. So if there are sigma factors, then there are also anti-sigma factors, and these inhibit where they bind. So it's a lot like allosteric inhibition, so it doesn't bind to the exact location that the sigma factor will bind to, but it binds to a location that changes the conformation of the actual site where the sigma factor binds to. The ECF is their extracytoplasmic function sigmas, and these are alternative sigma factors. They're very small, they're very simple, but they're incredibly diverse. And essentially, they're so abundant and they don't get used very often in biosynthesis. And so what Voigt and colleagues have done is that they've sort of used this in their circuits. So essentially in this paper, what they did was they created core orthogonal sets of 20 sigma to promoter pairs with minimal crosstalk. And this is really big because there have been circuits that have been made, but they never really work because there's always crosstalk going on, there's interference, and the circuits don't function all the way. So this is a really important thing for biosynthesis because they have been trying to make these circuits for so long. And basically, I'm going to go step by step how they made these orthogonal sets of the 20 sigmas and promoter pairs. So the first thing that they had to do before they really built the circuit was build their library. And they used the library by Ulrich and Zulin. And this is a library that was already constructed. It's called the MIST database, and it has over 19,000 ECF sigma factors. And then within that, Masher and colleagues have identified 43 distinct subgroups. And what Voigt and his colleagues did was they identified 2 representatives of each subgroup. So they built a library of 86 sigma factors. And this library is necessary because they're trying to use that library to design responsive promoters. So what they did was they designed a computational approach that identifies native promoters for each subgroup so that they can use the sequences to build subgroup-specific promoter models. The native promoters are really easy to identify because these ECF sigma factors auto-regulate and they can recognize their ECF sigma factors. So if it's upstream, the ECF sigma factors automatically know this is the native promoter that I need. So Voigt and colleagues created models that were used to screen whether the native promoters would be activated by sigmas in their own subgroups without crosstalk. They ended up identifying 706 promoters, and 29 unique promoter motifs for each of the 43 subgroups. All of these 706 promoters were then scored for their orthogonality, and they found that most of the promoters are very orthogonal and they have very little crosstalk, so there's little interference between the subgroups. And one of the most important things that they found within that is that the -10 domain and -35 domain, which is where the sigma factors usually bind, those are less orthogonal than the rest of the promoter. So it has a higher specificity that can be reached if you combine both of those regions. After they scored the promoters, they then decided to design their own candidate orthogonal promoters for the 29 promoter motifs that they found, that they identified beforehand. And they did this between the -60 to +20 promoter sequence. Basically, they selected the promoters that they predicted would score higher with their cognate sigma and poorer against others, because they are trying to reduce the crosstalk as much as possible. So they gave preference to promoters that immediately identified their ECF sigma factor. And of these, they found exactly 20 promoter cognates. Once they had these promoters and they identified these particular 20 promoter cognates, they had to measure the activity and the crosstalk that's created by these. So they sort of built a circuit, and they had an IPTG, which is what induces the ECF sigma factors, which can then bind to its particular cognate promoter. And once it binds based on its level of orthogonality, the GFP is fluoresced. So out of the previous 29 promoters that they had, 18 were functional with the sigmas from their cognate ECF subgroups. Once they had that, they could measure the toxicity and see if through the growth assays, and they realized that the sigma factors, none of them were toxic. And out of the 86 ECF sigmas, 58 activated at least one promoter at greater than 5-fold. So any sigma factors that were inactive usually showed non-functionality in both examples. So they had two representatives of each subgroup. If one showed non-functionality, the other usually did too. And this could just be because they got the motifs wrong, or if there is some property that they have that prevents them from being able to function in E. coli. So that is how they sort of measured the activity that was going on. And then in order to measure the orthogonality of the sigma to promoter pairs, they checked if the sigma factors altered the transcription of the host cell genomes. Generally, sigmas that are overexpressed were highly orthogonal, but had relatively orthogonal anti-sigmas if they were compared. And this is important if they wanted to build larger circuits. Instead of having that really small circuit of just the IPTG to the ECF to the promoter, they could build larger circuits. They noticed that there weren't any significant changes in the host genome expression through the transcription, so these ECF factors and these sigma factors don't affect the actual transcription of E. coli. And basically this supports that there is a lot of information content in the ECF sigma promoters, so that means that these won't affect the transcription of the host cell, and they can be placed in different areas without changing expression of the genes. While they made these libraries of the sigma factors, they then also had to make libraries of anti-sigma factors. And it was constructed much of the same way. They basically made a library of 62 anti-sigma factors as cognates to the sigma factors in the library produced beforehand. And of them, 46 of the anti-sigma factors associated with an active sigma. And of that, 32 of the anti-sigma factors could repress activity by greater than 2-fold. The problem with the anti-sigma factors is that they expressed toxicity. And before we learned that, the sigma factors showed no toxicity at all. So this could probably be just because of the interactions that the anti-sigmas have with the E. coli itself. And once they constructed this library, they could then test the orthogonality of these anti-sigma to sigma interactions by measuring the activity of the 32 anti-sigma factors that could repress activity before in the presence of the 20 sigma factors, the 20 sigma promoters that they had from before. So they found that most of these anti-sigmas specifically repressed their particular cognate sigmas, And this is really good because this shows that when they repress the cognate sigmas, that is the exact thing that they're supposed to do, and they don't repress the other ones that are around them. Once they have their libraries built of both the sigma factors and the anti-sigma factors, they could then assemble the orthogonal sigma factors, the anti-sigma factors, and their promoters into transcriptional switches. So they had an input promoter, which then induces the sigma, which then induces the cognate promoter. They found that most of the sigmas yield a graded induction of the output promoter, and what it shows that is that the output level equals the input level. And this is a problem that they found because then you can't have larger programs if they're equal to each other. So what they did to sort of counter that is they constructed a series of switches by controlling the expression level of the anti-sigma. And what this does is this increases anti-sigma factor expressions, and that will decrease the actual basal level, so the cooperativity increases. The basics of this paper isn't so much to prove something, but it's rather to expand on things that can happen in biosynthesis now. So because of the experiments that they've done, they've realized that the ECF sigma factors can be really easily moved between diverse organisms, but still be able to maintain their functionality. And then they showed sort of an overview of how bacterial genomes are organized in the ECF sigma transcriptional space. So if there is, basically if there's no crosstalk between the sigmas, then you have insulated expression. And the insulated expression leads to a particular preference. So you're gonna get that, that same sigma factor attaching with that cognate promoter repeatedly occurring. But then what they also noted was that if there is crosstalk between the sigmas, this could possibly allow multiple sigmas to cooperate in coordination, and this can lead to building more complex regulatory networks, such as having them cascade. So a sigma factor can induce another sigma factor, and go on. And it can also turn on multiple genes. And that's sort of the paper in a whole. Are there any, do I get questions or? Was that easier to understand? Was there anything confusing?
I thought it was good coverage. I had one question. I was wondering if anybody understood when they were doing this subgrouping, kind of what criteria they used for that. How did they come up with that?
The subgroups were actually already done by a different scientist, Masher, and then all they did with that subgroup was that they found 2 representatives of each subgroup, and that's how they built their library. So they didn't make the categories of these subgroups. The subgroups were already something that's in the MIST database.
Okay, do you know what they were based on?
I don't. I haven't looked at the MIST database, so I'm not 100% sure of how they based that.
The other question I had is, I was kind of surprised that when they put these into cells, that there wasn't any other interaction with the other cellular activity. And was that just they got lucky, or did they?
I think it has a lot to do with luck, to be honest. I'm sure they've done this repeatedly. ECF sigma factors are sigma factors that haven't been used a lot in biosynthesis, and this is sort of the first time that they're really using it. So since they don't affect transcriptional factors and they also have very minimal crosstalk, I think that they'll be used a lot more often in these biosynthesis circuits now.
I think it's by design because, first of all, there were a couple of sigma factors that actually interact with other cellular machinery, and with sigma 70, I think, or something like that, and they threw it away from the consideration. But also it's by design because ECFs are very specific, and they regulate the reaction of the cells for extracellular activity, for some shock. So they by design, by evolution, should be very specific and don't induce anything except some shock reaction. So I think that's why they picked ECF intentionally. And I think the grouping, if I remember correct, is mostly based on the motif of the promoter, the sigma factor target. So it's initially it's grouped by the promoter or by what it activates.
So just similarity in the promoter pattern then?
I think it's initial similarity in what it activates, what expression, gene expression it activates, and then they work backward by looking at the motifs before those genes and identifying the motifs by themselves. I think the databases group them by gene activations. If they activate the same genes, then they are in the same group.
Gotcha. Okay. Did one other question on the what is it, figure 1, that phylogenetic tree that they have? I was a little thrown off by that because I would have expected the different types of bacteria to kind of branch off at different points, and it seems like they show up at multiple points on that tree. Does that make sense?
Yeah.
Keep in mind that tree is only a very partial tree, right? So a lot of bacteria have multiple of these ECFs, and they've already selected a very small subset of them.
Okay.
So if they were to make the full tree, you might be able to see better the differences between the genes that have been evolved within the different lineages versus the ones that have been inherited between lineages.
Okay.
Sorry for the cool reverb.
It sounds very rock-starish.
Get back to the previous question about crosstalk with other systems in the cell, and just mention figure 4 is probably the most beautiful example of no crosstalk that I've ever seen. When you're doing gene expression studies, you're always expecting to see something that is near where you're affecting something at least. But in that figure 4, you can really tell that the 4 ECFs they picked really only affect those particular systems and nothing else. So they have very low noise on their gene expression data, and it's absolutely clear that nothing else is being affected.
So for your one other question on, so I hadn't really picked up on this, what is it, page 8, the threshold gated. I think that's where they're talking about kind of the gain of each system, is that right?
They're talking about what?
The gain, you're talking about it being,
Cooperative?
Yeah, where they're trying to increase that, and they talked about, essentially, if I'm understanding, they were using the anti-sigma to adjust the threshold, is that right?
Yeah.
Okay.
Because initially the input threshold was equal to the output threshold. And if they wanted to build larger systems, they couldn't if the thresholds were equal to each other. So they ended up decreasing the expression of the anti-sigma so that the thresholds wouldn't match up, and they could potentially then build larger systems, at least from my understanding of it.
And so just want to make sure I understand it correctly. They need only because on their plasmid the sigmas got expressed even without inducing the expression, and that's why they needed anti-sigmas mechanism to suppress that basal level, because basal level was created by uninitiated expression of sigmas from their plasmid, because their plasmid was leaking. Is that why?
I I think generally anti-sigmas are often needed as well because the ECFs are aren't they stress-induced based on environmental changes? So I think anti-sigmas still have to go in regardless of just to bind so that it's not binding all the time. But I do think that leakiness is a problem that they had with circuits before they used ECFs. And that was an issue of why circuits often wouldn't work. And so the anti-sigmas, I think, do stop a potential leakiness of a circuit.
Okay, but so let me rephrase. Where are those leaking?
Do we lose him? Hello?
And the ECF sigmas have helped with, at least through my understanding of it. The leakiness was the issues that they had with circuits before. And by using these ECF sigmas that they have now, the leakiness isn't there.
Yeah, the way I understood it was that they didn't have a leakiness problem. Their problem was they wanted absolute control of expression levels. So by having the anti-sigmas plasmid or change the levels of expression using the anti-sigmas, yeah, I don't think it was a matter of a leakiness issue. I think it was a matter of being able to control the system more thoroughly.
So my audio kicked out a little bit there, so I might have missed something, so sorry if this is redundant. But so what I was reading in this section is that essentially the basal expression of one stage could potentially trigger the next stage if you had two that were communicating.
Yeah, but where the basal expression is coming from?
I thought that was just that. I thought that's just the background expression. Maybe I have that wrong, but it's just kind of it. That's how it's expressed without a promoter or without a sigma factor.
So their plasmid got expressed regardless of sigma factor that makes it expressed, because they designed a plasmid that got expressed when you add lactose or something like that. So, but if I understand that correctly, their plasmid expressed sigma factor regardless of adding anything to the mix. So to suppress that noise, they added anti-sigma. So my question was where those noisy basal level sigmas coming from? That's
somebody else. Yeah, somebody else might be able to jump in, but I thought they were.
There's the T7 promoter, which might be leaky, and then there's the actual ECF promoter that might be leaky. I'm not sure whether they clearly distinguished which one was at fault there. One thing I was wondering regarding sort of improving on the switches towards the end of the paper was why they didn't try to have the ECFs be auto-regulated, because that's an easy way for getting a response curve, is to add positive feedback essentially, right? So you have the ECFs being regulated by their own clock promoter, and then you'd have to have some actually switching on that system, but that is something you could then do with the anti-ECF.
So the analogy in electronics would be kind of a circuit that would either force it high or force it low, rather than just having kind of the absence of a high signal.
So you make a bistable switch essentially, right?
Right.
That's what they were complaining against, that you had a two continuous response rather than just having an on-off behavior.
Mm-hmm.
Yeah, I guess that they just didn't test it because, and also if you make it auto-regulated, you make it really sensitive. Probably that's why they didn't do it, just to not induce the noise.
But again, the input for the system in that case could be the level of the anti-sigma, right? So if the anti-sigma goes high, then you switch off that bistable switch. So it's another way of doing it. It'd be interesting to see what difference it makes compared to what they did. I was wondering whether it might be useful to take a step back and talk a little bit about where these ECFs come from and what the terminology means, and what these anti-sigmas really do in a cell. So I don't know, I came in a little bit late. I don't know if you covered that in the beginning. So the reason why these are called extracytoplasmic function sigmas is because they typically work with together with one of these anti-sigmas that is typically a transmembrane protein, right? So you actually have a protein that sticks through the membrane and receives some signals from outside the cell, and then it has a domain inside the cell that interacts with these sigma factors, right? So typically, when it doesn't receive any kind of signals from outside, the domain on the anti-sigma that's inside the cell will actually bind the sigma factor and sort of sequester it and prevent it from interacting with the DNA. But then when it receives a signal from outside the cell, the sigma factor is actually released from the anti-sigma and can then interact with the DNA. That's why they're called extracytoplasmic function sigmas. They're actually sensing something outside of the cell and then conveying that to gene expression levels inside the cell. And so normally these anti-sigmas are actually 2 or more domains. And here in this paper, they're really only focusing on the domain that interacts with the sigma factor. Right. So, when you start looking for these proteins in the databases, you won't just find what they call the anti-sigmas here. You'll find much larger proteins that include one of these anti-sigma domains. I just wanted to put it out there because I think it's kind of confusing to talk about these extracytoplasmic factors, and it's kind of hard to understand what they're actually doing in nature.
Can you give examples of those external factors? I know only one is a temperature. What else? I'm just curious.
Virus.
I think pH is also a factor.
What? No. Well, what external factors triggers those sigmas, cellular sigmas, to be activated in nature? What functions they play? I'm just curious.
I think, yeah, that's what I was saying. I think that a change in pH may also change it and change what it does, along with temperature changes. We're not really sure of other ones. Those seem like really big changes in the external environment that might change how it functions.
There's a couple of nice reviews in the initial reference in the introduction, talking about these ECFs and what roles they play in various bacteria. I actually printed it out and then forgot to bring it, but they have a bunch of examples of what they interact with. So just from memory, I think they interact with a range of small molecules. So they tend to be fairly specific. So yeah, whatever signals you can imagine, like a 2-component system, for example, might sense. They kind of act in similar ways.
So it seems like one of the hopes of using a ECS system like this is that you could selectively turn on and off expression levels. The small molecules you're talking about, Patrick, are they specific enough and with a few enough other side effects that that you could do that in a living system?
I would think so. So that's an angle that they completely don't explore in this paper, but some of these other reviews do look at that. Use these systems as actual sensors rather than just building the logic inside the cell.
I'm wonder if you can get one of those anti-sigma proteins that embedded into the cellular membrane and just change the domain and make it repress a different sigma that's orthogonal to everything else, one of this paper, and make it control some of the internal machinery. So that way you can control the expression of your plasmid. Just an idea.
I think they did something similar to that, right? With the chimeric sigmas, where they took the different domains. So they took the -10 domain from one cell and they took the -35 from another.
No, what I mean that let's say that there is a anti-sigma embedded right now into the membrane of E. coli that reacts for heat and release some particular sigma. We can get a DNA sequence for that anti-sigma that reacts for heat and replace the domain with the domain of another anti-sigma that is not specific to E. coli, one from this paper, and make it controlled and a sigma that affects our plasmid, that we injected in the E. coli. That way we can make E. coli react on heat and activate the plasmids that we put into it. Is it super confusing what I'm trying to explain?
No,
that makes sense.
That's why I'm asking what else you could do to, what other signals it reacts for. I'm just curious, what the less harmful thing you can do to the cell to activate those, anti-sigma. So I could use that mechanism to reprogram it to activate something else.
Yeah, I think that's entirely possible. The review that I was talking about is held on 2002. The extracytoplasmic function sigma factors. I'm just looking through it right now. They have a whole section on sort of figuring out functions of these factors. So in E. coli, you have sigma E, which is periplasmic stress, extreme heat shock. There is one that's involved in ferric citrate uptake system. Let me just browse through this a bit more.
There's also the MIST database online too.
Yeah,
that looks interesting. I'm just surprised that this isn't used more often for inducible expression.
I think the paper just came out, so it might be a while before it starts getting adopted. As far as I understand.
Yeah, I think the whole purpose of this paper that they identified the orthogonal sigma factors, and before probably people knew about all the sigma factor and anti-sigma factors, but they couldn't use them because when you activate them, and they also activate the big number of other promoters. But with this orthogonal sigma factors, you can redesign the existing anti-sigmas just to work for you, only for your plasmids. That's I think the cool part of this paper.
Also,
Right.
I'm not sure how much is actually known about the specific signals that these things sense in the environment. Looking at this review paper here, it seems like there's more essential functions of the genes that are being regulated. So they just look at the regulome affected by that sigma factor, and then they sort of put the function of that on there. So just looking through this list, you have like disulfide oxidoreductase. That's probably sensing some kind of sulfur compound, but they don't specify what. There's a number of proteases. Those might be recognizing specific peptides potentially. There's lipopolysaccharide biosynthesis. So that's LPS recognition. They might be recognizing maybe other bacteria based on the LPS on their surface. So yeah, there's lots of interesting functions of genes regulated by these sigma factors, but I don't see a corresponding table of the actual signals that are being recognized by the sensors.
Yeah, something I wonder if the goal here is to modularize all your parts, then you wouldn't want a sigma factor or an anti-sigma factor that is related to a specific signal. You'd want that to be somewhere else in your circuit. At least that's the way I would think about it.
But the sensor is really the part that sticks through the membrane, right? So that is, and they're completely separate that here from this analysis, where they're only looking at the domain that's interacting with the sigma factor, right? So there's a sensor part, that is the transmembrane part of the anti-sigma. They're completely ignoring that part here. So it's kind of interesting. So for a long time, there have been these two camps in synthetic biology with regards to how to make larger circuits. I always make the joke talking about synthetic biology is that we're sort of at a stage where if you compare it to the computer revolution, we're still at the stage where we're all making our own transistors from a chunk of germanium. And we really only have 3 good wires to work with. So there's here. And there's 2 different ways of really going about that. One is to use the existing transcription factors that nature is using. And if you're doing that, then you have to worry about how orthogonal these things are. Right? Can you really getting a large enough collection of these transcription factors that you're not going to get crosstalk? The complete alternative to that is by taking advantage of sort of the addressability of the DNA itself and using more systems that are based on riboswitches, where you can actually express a piece of RNA from one gene that then start affecting the expression of another gene at the RNA level, right, post-transcriptionally, by messing with a riboswitch that prevents or induces translation of the second gene. So that's a completely different approach to making more wires for synthetic biology. I had always expected that approach would have been far more fruitful in the end, because it's essentially infinitely addressable, right? You make you have a tag that says where this wire should connect to. The tag is essentially a DNA sequence. If you're always doing it based on existing transcription factors, then you have to keep mining nature to find more and more wires that you can use that don't interfere with your other wires. But papers like this really show the potential of doing that and actually coming up with combinatorial approaches to making more and more wires.
What did you say that the RNA approach, what did they call that?
Riboswitches.
Riboswitches, okay.
Right? So you would have a 5' untranslated region in front of the actual coding region of the gene. And that 5' untranslated region forms a structure in the messenger RNA that either activates translation or inhibits translation. And by binding another piece of DNA or RNA to that RNA structure, you can make it fold or unfold, essentially. Right? So you can regulate the expression of the gene at the translational level by using another piece of DNA. Right. So if you want to build a big circuit that says, I want to transmit some kind of signal from point A to point B, you can either do that by expressing some kind of a transcription factor that finds its cognate promoter and activates that promoter, or you can express a piece of DNA or RNA that affects the translation of an already expressed mRNA.
So Patrick, what's the advantage of a system like this versus a system like that?
Well, so a system that's based on ribose switches and addressability by RNA sequences is that you can code it very easily, right? If an RNA sequence of length 5 isn't specific enough, you make it length 6, or you make it length 7 or 8 or whatever. So you can always make more wires essentially by addressing those pieces of DNA more and more specifically. The advantage of building something based on existing transcription factors is that they're already available. You just have to mine for them, and once you find a good set that's orthogonal to them, anybody can PCR those out of the organisms that occur in, and you can start using those. It requires a little bit less thinking, perhaps, because really to take advantage of these ribose switches, you have to be able to design in interaction between multiple ribose switches in front of the same gene, and it gets a little hairy. But I think some of those problems will probably get resolved fairly soon as well. There's quite a few people working on ribose switches and actually building sensors directly in ribose switches. You can make mRNA hairpins that have an affinity for small molecules, right? So depending on whether the small molecule is present or not, the hairpin will fold or will not fold, and the downstream gene will get translated or not. So there's all kind of cool tricks you can do, not just sending a signal from point A to point B, but actually building in the sensors themselves as well. Anyway, but that's drifting away from the current paper, of course.
Where do you see the specific technology going, out of curiosity? This particular, using sigma factors. I haven't seen this before.
Well, it greatly expands the repertoire of transcription factors we can use, right? So if you look at the other pieces that you're using in this paper, there's like a T7 promoter, there's IPDG inducible promoter, there's one other one, I think. Those are the standard ones that everybody's using, right? And there's really only a handful of those that every single IGEM project uses to send signals from point A to point B. If you want to make one of these oscillator systems where you have 3 genes that all repress each other, if you want to make that any larger, you're going to start running out of repressors and transcription factors to regulate the genes involved. So this kind of paper really hugely expands the repertoire that we can use to connect some of these logic pieces of a larger genetic circuit together.
And if I understand correctly, using anti-sigmas allows you to control the different concentrations of different sigmas. So you can, if those segments control the expression of particular proteins, you can build a circuit where you can control the level of expression for every protein. You can have one expressed 5 times more than another one using these sigma factors. So you can achieve the required concentration if your system requires a particular ratio between concentration, if I understand that correctly.
Yeah, tuning these anti-sigma factors, I don't think it's all that straightforward. You would essentially have to put anti-sigma factors behind constitutive promoters and then tune their expression level to whatever specific set point that will give you the correct behavior for the sigma factor circuit. Right. So it's a little bit tricky.
Yeah, but at least they give you knobs.
Yeah, exactly. Yeah. But there is some infrastructure that you have to build for each knob as well. Right?
So, Patrick, can I suggest that some of those links to some of that we add that on to the website? Then we can put the MIST database link. If you have something on ribose switches, that'd be interesting to throw on as well for anybody who wants to follow up.
There's a couple of really cool papers on ribose switches that we can maybe pick out for another journal club.
Okay.
I'm probably not the best expert on ribose switches, but there might be somebody else here that is.
Best doesn't have to be extremely high.
Brad, I'm just curious, do they have as many switches? I mean, right now they have 20 switches here for the sigma factors. Potential to grow to, I forget the number, something like 100.
Oh, that can grow to a huge number because you can get -10 and -35 domain from every sigma factor and flip it with every other one. You have a huge combination. It runs in hundreds easily.
And they've only tested 2 examples from each of these subfamilies, right? So it's thought that these subfamilies tend to share the same -10, -35 regions, but I'm not sure how certain that is, and I'm not sure if they've done a good analysis in this paper, if they picked multiple promoters from the same subfamily, whether they really have identical affinities. I guess some of that is in one of the papers, right? Let me see. There were some really good additional supplementary figures.
Yeah, if you look at the figure 2, you will actually see that the promoters motifs for every subgroup is pretty strong. I mean, they're similar. So
yeah, but I'm wondering how different the promoters are within the subfamily. And I guess you can see some of that, or I guess how much promoter specificity is. If you, I don't know, if people have the supplementary information open, there's a figure S4, which is the full version of figure, let's see, figure 3A, I think. So figure 3A shows you that nice diagonal matrix with the sigma factors affecting the different promoters. So in the supplementary information, you have the full matrix, right, including all the crosstalk. And there you do see that there's so the number of rows, you have 2 rows essentially for each of these subfamilies, right? Because you have 2 sigma factors that they picked from each family, subfamily, whatever. And they do seem to behave fairly similar in terms of how they interact with the promoters and what kind of crosstalk they have with other promoters. So I guess, yeah, within the subfamily, you do have, they seem to be fairly equivalent to each other within the subfamily.
Yeah, because they build the promoters based on the promoter motifs for the whole family. The promoters they use, they don't exist in nature. They synthesize the promoter sequence by taking the most common nucleotides for all the motifs for the family.
No, I think they actually picked one. They picked an existing promoter.
Now, if you look at back, Well, I think. Oh, you think they picked the one, but they used the model to scan it? Okay, I'm
Yeah,
not.
I think that's what they're doing. Yeah, that was my understanding. So there might still be some room for additional diversity to be found by looking within these subfamilies, but probably not that much more than what they show here. Uh,
But that's still a lot. 20. Can you think of synthetic biologist circuitry when you will need more than 20 or even 10 orthogonal promoters?
That's like that famous quote saying, I don't think we would ever need more than 10 computers across the world, right? Or whatever. I don't remember the exact quote, but yeah. I'm sure we'll get there very quickly.
Right.
No,
So
but
they say that on page 6 on the right-hand side, they basically say that they estimate that using the technique that they found, they can probably pull up about, and switching the different motifs, they could probably make about 160 potential sigmas.
Yeah.
So that's a little more than 20, but again, it's not infinite.
And not all of those are really going to be orthogonal, right? You do get some crosstalk. And actually, if you compare that figure in the supplementary information with the nice orthogonal figure in figure 3E, you'll see that there is quite a bit of crosstalk. These things are not quite that specific as they make it seem here. They're just
no,
focusing on the ones that
no.
are the most
It takes
orthogonal.
in account the crosstalk. I'm sorry. It takes in account crosstalk. They take in really diverse -10 and really diverse -35 domains, and they saying that we have 160 combinations that should be specific. You know, because if you don't take that in account, you should get 400 of them, 20 by 20.
Right.
Or I think 200 is to be correct, but anyway, they take that in account.
Sorry, I'm going to cut in. I actually have to leave. Thank you for having me, though. I hope your discussion continues. It was really nice, and I think I have a better understanding of the paper now.
Well, thank you very much for your time. Thanks for presenting the paper.
Yeah, thanks for the paper.
Thank you. Thank you.
I'll go ahead and stop the recording then.